Hello, and welcome to Inside the Alzheimer's Lab, brought to you by Cure Alzheimer's Fund. I'm David Shank, author of The Forgetting and senior advisor to Cure Alzheimer's Fund, a not-for-profit scientific consortium dedicated to ending Alzheimer's as soon as possible. We bring you a series of these discussions throughout the year to help improve public understanding of the disease. Today we're going to be talking with Dr. William Mobley, an expert on the relationship between Down syndrome and Alzheimer's disease. And we'll be taking your questions as always. Please submit them to our Facebook page at facebook.com/curealzheimers. William C. Mobley is a distinguished professor and chair of the Department of Neurosciences at UCSD. He also serves as executive director of UCSD's Down Syndrome Center for Research and Treatment, and is the Florence Reifert Chair, sits in the Florence Reifert Chair, I should say, of Alzheimer's disease research. And he's also a member of the Cure Alzheimer's Fund Scientific Advisory Board. And there's another page full of outstanding credentials we could read, but that would take up most of our hour. So I'll just jump in and welcome Dr. Mobley to our program. Thank you so much for taking the time today.
My pleasure to be with you, David.
Dr. Mobley, let's talk first about how you got into both Alzheimer's disease research and Down syndrome research, taking that in any order that it makes sense.
Well, you know, this really started when I really fell in love with proteins and how proteins work as an undergraduate at the University of Nebraska, and found this wonderful opportunity to work at Stanford University with Eric Shooter, and with Eric to explore the biology of a new protein called nerve growth factor, which is really important for keeping neurons alive and well. And as it just happened, I was leaving the residency in neurology at Hopkins, and this protein hit the big time because it was discovered to be active on brain neurons. And that was really not just that, but the idea that it might work on brain neurons that were threatened in Alzheimer's disease made it very exciting to think about committing my career to understanding how to really know about Alzheimer's disease and treat it effectively. The connection with Down syndrome really was through Alzheimer's disease, because we knew even then, in the 80s, that everyone with Down syndrome develops the neuropathology of Alzheimer's disease. So the connection was protein to nerve growth factor to Alzheimer's to Down syndrome.
Now we're going to be talking about some of your recent research and how that connection is better and better understood and what the implications of that are. Maybe you could first generally characterize how what the body of knowledge has been in the connection between Down syndrome and Alzheimer's since you started to work in both over the last 10 or 20 years.
Yeah, that's great, David. So when I started this, I think we knew for sure that Down syndrome was really very much linked with Alzheimer's disease. But the questions remained: how close is that linkage? And what explains that linkage? And how could one, knowing that that link existed, really move to understand how the disease emerged and what to do about it therapeutically? When we started, we knew that the pathology was there, but we really didn't know how that emerged. And over time, it's become clear that it's an extra copy of the gene for APP that in large part determines whether or not people with Down syndrome get Alzheimer's disease. So this is this protein that's encoded by a gene on chromosome 21, and when you have a perfectly normal gene but present in one extra copy, it's necessary in Down syndrome for Alzheimer's disease. So say it differently. People with Down syndrome who miss out that extra copy of the gene for APP do not develop dementia and do not develop the neuropathology of Alzheimer's disease. So that's a very critical clue. And so, yes, it's gene dose for APP, and then what does that mean? And in recent studies, we think we understand now how that extra copy of APP acts, and we think that the mechanism by which it acts is very similar, if not identical, to the mechanism by which other people get Alzheimer's disease.
So we're going to go into even more detail in just a second. And before we do, let me just back up and talk about, just on the people side of this, the original just observed link, general link between Alzheimer's and Down syndrome. Is that, can we say, is it almost everyone who has Down syndrome eventually does develop dementia? How, I know it's a very strong link there, but from the clinical side, what is that? What are the numbers look like?
It's important, and I would argue that it's a point of, I wouldn't say it's controversy, but the ways people speak about the data differs. So what everyone agrees at this point is that if you have an extra copy of the gene for APP, that is almost certainly necessary for you to develop the pathology, the brain changes of Alzheimer's disease. What people are still trying to come to terms with is, so if you have the pathology, does that mean you have dementia? And the answer is no. Just because you have the pathology doesn't mean you have the dementia. But it is the case that by age 60, if you have Down syndrome, you have a more than 60% chance of having dementia in addition to the brain changes. And the estimate is that if you live long enough and you're a person with Down syndrome with an extra copy of APP, you will develop dementia. Now that's probably stating it very strongly, but I think the data is there for us to look at. And in essence, like Alzheimer's disease, this disease progresses over time. So the brain changes are likely to predate by a decade or two the dementia that finally ensues. So I think it's very right to say that not everybody at age 60 has Alzheimer's disease, dementia, Alzheimer's disease like dementia. It's right to say that they have the pathology without dementia. And it's also right to say what is different about them, what other genes might be acting, what other environmental influences might be acting to prevent Alzheimer's disease in those folks. Having said that, I think the data is also very strong. If they live long enough, these people will develop dementia. And they are living longer. I should mention that, you know, in the first part, in the early 1900s, the average age at death for people with Down syndrome was less than 10. It's now at least an average of 60, and it's very likely to go higher. And why would that be? It's because of advances in medicine. It's advances in medicine that are made available to people with Down syndrome. So now, you know, they get antibiotics for pneumonia. They're treated for these medical disorders and surgical disorders that they weren't treated for before. And importantly, young children that have a much higher incidence of congenital heart disease are now operated on, and very successfully. So now that whole population, like the rest of us, is living longer.
Thank you for that clarification. So you brought along some slides. We asked you to bring along some slides, and we're gonna invite you to really climb into to your research in some depth, and we'll ask for forgiveness from the general audience if it gets a little bit technical, but that's part of the point of this program, is to give people really a visceral sense of what is going on inside these Alzheimer's labs, and to get to know the scientists and the nature of their work. And we talked about this earlier, but I may or may not interrupt you from time to time to ask you to clarify a few things. But for a few minutes now, you're going to go through some slides and get into the meat of your current research.
Sounds great.
I could ask the technical producer to bring up the slides, and hopefully you will see those. I think we now have the first slide up, introducing you and the slides, and I suppose we'll want to go to the next slide to.
Let me just, so my vision, so my version of this is tiny. And is there a way that I can make it bigger, or do I just live with what I got here?
That's a good question. If you click on the icon, I'm told, there's a good chance that it will get larger. Tell us if that works.
Yeah, perfect.
Okay, great.
Well, it was perfect for a second. Okay, can we go to the first slide then, if we could?
Sure.
Again. And this is just to introduce the theme. And so the theme here essentially says we could now, because of what we know, especially what we know over the last few years, we can now rethink Alzheimer's disease, its cause, and its treatment in the context of Down syndrome. So the presentation is really aimed at informing the audience of the potential benefit to understanding and treating Alzheimer's disease, of understanding and treating Down syndrome. So that's the main thing. Next slide then. I put a slide in as a kind of glossary. If we can go to the next slide. I wanted to make sure that people knew the meaning of words I will use a lot. And so pathology, by that I mean the changes that can be seen in the brain. Technically it's neuropathology, but we'll just use the word pathology. So these are the changes in cells and neurons and other cell types that one sees when one does cuts sections of the brain or looks at the brain and examines them. So that's pathology. Pathogenesis is a different word. It's a word that essentially says this is the cause of the disorder or disease. In other words, what's going on that makes this disease happen and what makes it worse. And here, as I say there, one's usually speaking about a mechanism. So how does it happen that an extra copy of APP makes Alzheimer's disease far more likely? And by sporadic, I'm basically distinguishing cases that occur without a family history to cases that occur with a family history. We call the ones with a family history as familial Alzheimer's disease, or FAD, and those that don't have a family history, we call them sporadic. It's absolutely true that most cases are sporadic, and so one has to remember that there are different categories of disease of folks who ultimately are diagnosed with Alzheimer's disease, some in families, some with Down syndrome, and some with so-called sporadic disease. So let's look at the next slide.
Interrupting 1 second. Are we saying that the people with Down syndrome who develop dementia, do we count those as cases of sporadic Alzheimer's or familial?
Neither, because it's not in their family. Okay. And we have a genetic cause for it that would make it not equal to sporadic. So it's a third category, if you will.
Okay, I appreciate that.
Sure. Okay, and next slide then. Good. So this is a very highlighted version of the pathology, the brain pathology of Alzheimer's disease and Down syndrome. So like Alzheimer's disease. Now we're looking at the brain on the left-hand side of the screen. On the left-hand side of the left-hand side is a healthy brain, and notice that there's there's a cortex which is stained brown here. There's lovely white matter that's white. The brain is nice and full. The sulci are coming up against one another. There's very little extra room. When you look at that brain, it's tightly packed. On the right-hand side of that, this is a brain, a half of a brain of a person with advanced Alzheimer's disease, and notice the brain is shrunken. The brown-stained areas of cortex are thinner. The white matter is less. The ventricle, that one sees that space, is much larger. And now look toward the bottom. And toward the bottom, I wish I could point to it, is a structure very near the midline that's called the hippocampus. It's the part of the brain that allows you to learn new material for events and episodes. It's episodic memory. That is robust and beautiful in the healthy brain, but in the advanced Alzheimer's disease brain, that space, that little circular space that's between the white matter and the brown stained cortex is markedly enlarged, and it's enlarged because the space was once filled with hippocampus, and now that hippocampus is shrunken. And that really fits very well with the idea of memory loss, short-term memory loss in people with Alzheimer's disease and in Down syndrome. Now, if we look in the middle, here's another very characteristic part of the biology, of pathology, if you will, of Alzheimer's disease and Down syndrome. This is called a neuritic plaque, and the plaque features a couple of elements. The central element there is a batch of protein, a spherical collection of a peptide called Abeta. We'll come back to that. The other brown stained material around there is partly the Abeta peptide, but it's invested with these very dark stained little processes, and those are both axons and dendrites. Let me say a little bit about axons and dendrites. There are three parts of a neuron. There's a cell body, which contains the nucleus. There's a thing that's like an antenna. It receives information. It's called the dendrite. And there's another piece called the axon, and that is the element that comes out of the cell body and goes to the next cell, next neuron in the circuit that this neuron participates in. And so dendrites are connected to axons, or say it differently, the axon of one neuron is in contact with the dendrite of the next neuron in the circuit. Both of those elements, the dendrites and the axons, are present in these plaques, these A-beta peptide containing plaques. Now, on the right side, there's another thing called a tangle. And tangles are, again, misfolded proteins that represent essentially the accumulation of a particular protein in neuron cell bodies and dendrites and axons in the brains of people with Alzheimer's disease and in people with Down syndrome. And we know very little of what we'd like to know about plaques and tangles, but we know that they're absolutely classical features of pathology of Alzheimer's disease, whether one has familial Alzheimer's disease, sporadic Alzheimer's disease, or is an elderly person with Down syndrome. And I say on the bottom, the pathology of Alzheimer's disease emerges in everyone with Down syndrome by age 40, with dementia in greater than 60% by age 60. And of course, that has to be conditioned by saying that if you don't have that third copy of APP and you have Down syndrome, you don't develop this pathology. But by and large, this is the case. One sees the pathology in essentially every person with Down syndrome by age 40, and most have dementia by age 60. So let's go to the next slide. So this is the if-then proposition for Down syndrome and Alzheimer's disease. So if there are fundamental links between pathogenesis, that means disease causation in Alzheimer's disease and Down syndrome, and one can define those links and their mechanisms in Down syndrome, then one could integrate work in Alzheimer's disease and Down syndrome to diagnose the earliest onset of AD pathogenesis. Remember, we have a population that we can identify as ultimately contracting Alzheimer's disease on the first day of life, even in utero. And if we could do those things, we could build a more robust ability to develop and test and validate effective treatments. So this is the if-then proposition. I think it's an exciting one because it lays out the possibility that by understanding Down syndrome and the age-related changes that are like AD, we could understand both better. So let's just go through these if we can very briefly. What about the first one? If there are fundamental links. And so the next slide talks about these fundamental links. So as mentioned before, the brain pathology is very similar, if not identical. Work for Brad Hyman and his colleagues in the mid-90s, very convincing in this regard. It isn't just that the pathology is similar, it occurs in a similar regional distribution and over time. So very convincing. As mentioned, it proceeds over decades. A person with Down syndrome will have the pathology of Alzheimer's disease 10, 20 years before they develop dementia. And that's like Alzheimer's disease, where we know that there are early changes in processing of APP and in the brain, volume of the brain, elements of brain pathology, biomarkers present literally a decade or two before the onset of clinical memory loss. And finally, this disease causation, this mechanism implicates APP, this amyloid precursor protein, as it does for Alzheimer's disease. So, as mentioned, the genetics of AD that happens in families implicates APP, either through mutations in the protein or through mutations in enzymes that process that protein. One should remember that there's an extra copy of APP in some families. It's a perfectly normal copy, normal gene, but it actually is sufficient in and of itself, this extra copy, to cause Alzheimer's disease, pathology, and dementia in families. We know that there are changes in the processing of APP in sporadic cases. And finally, as mentioned before, deleting APP or not having that extra copy of APP prevents brain pathology in Down syndrome. So I think we can make a very strong case that the pathology in the brain is similar, that the time course of changes in the brain are similar, and that APP plays a central role in both typical Alzheimer's disease, that's sporadic, familial Alzheimer's disease, and in Down syndrome. Next slide. Okay, a really interesting piece of the pathology that we focus on is the endosomal pathway. And we were very much educated about changes in endosomes by Randy Nixon and his colleagues, and Elliot Mufson, and Ann Cataldo. And what I'm pointing to here with the top red circle is this very important compartment called the early endosome. Now, what are endosomes? Endosomes are the following. The cell is sitting in the environment in which it lives, and it's constantly taking in nutrients. And what it does is to pinch off a bit of the cytosol, pinch off a bit of the extracellular world, pinch it off, and carry it inside the cell. And that's what is going on in the upper left-hand corner. There's a little circle. It's pinching off a bit of that white space, that is the extracellular fluid, and it's carrying it inside the cell. And one used to think that that was just about getting rid of surface receptors that were no longer needed. But we know now that that little red circle over the early endosome, we know that those early endosomes are terribly important for carrying trophic messages from the tips of axons to the cell bodies that make those axons, and that those early endosomes are playing a role that's essentially teaching neuron cell bodies that they do in fact have a target, they should stay well, they should make the right genes and proteins and stay functional. If we could go to the next slide.
And I should just point out here that it's what we're seeing is just a technical lag. When you call for the next slide, it just takes a few seconds for it to come up.
So now, why am I so interested in early endosomes? It's because they carry those messages that are trophic messages. And so what you see here in the upper panel is a blue neuron, cell body on the left, axon in the middle, and axon terminal, synapse, if you will, between the axon and that gray dendrite. That diagram is shown to you because, as you may be able to see, one's forming these early endosomes that carry the trophic factor bound to its receptor. That's better seen below. So that little tiny circle in the blue axon is blown up in the bottom of the diagram, and notice that there's this early endosome, has lots of proteins attached to it, but very importantly inside it, it's a growth factor bound to its receptor, and that growth factor bound to its receptor is vital for the life, survival, and function of that blue neuron and of its connection with that gray dendrite of the next neuron in the circuit. When you disrupt trafficking of these so-called signaling endosomes, you disrupt trafficking of trophic support, and our evidence is that if you do that, this neuron, the blue neuron, shrivels up and dies, and is very dysfunctional before doing so. We think this is a critical part of the pathology of both Alzheimer's disease and Down syndrome. Next slide. So here makes the case that in Alzheimer's disease and in Down syndrome, one has interrupted that flow of information and has compromised the well-being and function of the blue neuron and the circuit in which it participates. Now we'll talk about how that happens in just a second, but let's go to the next slide. So let's examine the second proposition, if you will. We've already seen that there are fundamental links between pathogenesis, between the cause, between the pathology of Alzheimer's disease and Down syndrome. So can you now better define how that pathology arises? And we now define the underlying mechanisms in Down syndrome? So the next slide will very briefly sketch what we know. And we know this because we've been able to use mouse models of Down syndrome. These are genetic models in which one has an extra copy of many of the same genes that are responsible, that are on human chromosome 21, and that appear to be responsible for Down syndrome on human chromosome 21, including the gene for APP. These are the TS65D in mice created by Muriel Davison and studied so nicely by so many people in the Down syndrome research community over the past, really now 20 years. Important contributions by people across the world, including work at Johns Hopkins and Arizona and Stanford and other leading institutions in Europe, including investigators in Paris and Italy and the United Kingdom. Important laboratories studying these mice in London at University College London. So next slide. What we learned is that that little mouse teaches us the following. That is, it's the extra copy of the APP gene that causes an increase in the levels of its protein and the breakdown products of that protein. These mice show age-related dysfunction and death of neurons that are classically involved in Down syndrome and Alzheimer's disease. The death of these neurons is correlated with changes in synaptic and axonal function. In all cases studied to date, increased APP gene dose is responsible for degeneration, and, very importantly, extra copy of APP causes the same kind of enlargement of endosomes that one sees in Down syndrome and Alzheimer's disease. And transport is defective, and so if we could just go to the next slide, I'll show you something very interesting. Now, this is several years old. When we studied the transport of this protein that I referred to before as nerve growth factor, NGF, if we put radiolabeled NGF in hippocampus, we know normally it's transported to neurons that are vulnerable in Down syndrome and Alzheimer's disease, and when we put it into a normal mouse, the transport over the label is 2N, very robust transport, certainly capable of keeping those neurons alive and well. But look at the dramatic decrease in transport that occurs in the little mouse model of Down syndrome. It's 5 to 10% of normal. And we would regard this as a critical defect in transport that's very likely to lead to atrophy and death of these neurons, as we see in this mouse model. What we can say is that that transport is markedly improved if we just knock out one copy of APP in the mouse model, just one copy of one gene, the gene for APP, and now these neurons, these cholinergic neurons, no longer degenerate. So APP clearly plays a critical role, not just in Down syndrome, but in this mouse model of Down syndrome. Next slide. And what is it about APP? This schema shows you how APP is processed. Here's the protein sitting in a membrane of a neuron. So right in the middle with the big letters APP, that's the full-length protein. Now let's focus on the right-hand part of the screen. It's susceptible to cleavage by this little symbol called beta. That represents beta secretase. When beta cuts, if you just move from the middle one step over to the right, you see the release of this thing called APPS beta, and you see the presence of a thing now still in the membrane called C99. Now, if the enzyme called gamma works, another step over to the right, you see the release of A beta and the release of something called AICD. What we can say in recent studies in vitro is that those three arrows mark each of the products of APP that are capable of causing endosomal dysfunction and neurodegeneration. The full-length protein, C99, and A beta all cause misery. What doesn't cause memory is C83. If you look over on the other side, that doesn't cause a problem, nor does AICD cause a problem. So I think we're narrowing down what it is about APP gene dose, and that is that there's too much of the full-length protein, there's too much of the C99, and there's too much A beta. That, we think, is critical to pathogenesis in both Down syndrome and in Alzheimer's disease. Next slide. Now, I've mentioned that this transport doesn't go well. That on the left side is the normal situation where we've got nice little circles flowing back to the cell body. That's very different in Down syndrome, where there are fewer of those little circles getting back, but notice that they pile up. And they pile up in the synapse at this point of contact between the axon and the dendrite. And we know now from studies done recently that this is another problem for the neuron. It's not just that the cell body is deprived, it's the synaptic contact is too active. There's way too much signaling of trophic factors, very likely important for synaptic dysfunction. So it's kind of a double trouble, if you will. And we think this is true not just in Down syndrome, but in Alzheimer's disease, the latter yet to be proven, but we're very suspicious that the changes in APP processing are responsible for both changes in the synapse and in the cell body. Next slide. So in schematic form, and this is now specifically Down syndrome related, an increase in APP gene dose leads, in the third arrow, to an increase in endosomal size, a decrease in transport of these endosomes that carry these trophic messages, so that there are changes at both the synaptic domain and in the cell body, with resulting neurodegeneration. So that's the overview of what we're doing, and we think there's a lot to learn from this pathway to really understand the mechanism by which neurons get sick and die and how to fix them. Next slide. Now, what about the third proposition, that if these things are true, if there's a nice correspondence in pathogenesis, and we can explore that pathogenesis in models of Down syndrome to derive new insights, what might we do to study the Down syndrome population with these insights to help all the rest of us who are susceptible to having Alzheimer's disease? So here are the apparent benefits of engaging the Down syndrome community. First of all, this is a cohort at a markedly increased risk for Alzheimer's disease. The folks are easily identified by their facial features and their physical features. We know that the pathology is there and that we can make the diagnosis decades before the onset of dementia. In the main, this is a pretty healthy group of people. We think there's about 150,000 people in the U.S. between the ages of 30 to 60, and we've encountered caregivers that are highly motivated to make a difference in their loved ones' lives and are willing to participate in clinical research and in clinical trials. And recently, under the NIH, there's a registry that's been established and enrolling under the Down Syndrome Consortium. So this is a population of folks with caregivers and loved ones and siblings and mom and dads that's large and eager to participate. Importantly, this group allows us to examine the pathology and the pathogenesis longitudinally. We can begin in 30 or 20 or 15-year-olds to explore what's really going on. And these folks would then be available to construct not just trials, but trials that aim at primary prevention, preventing Alzheimer's disease from occurring at all, as opposed to treating symptoms or preventing symptoms from becoming more severe. So we think studies of Alzheimer's and disease and Down syndrome make a lot of sense. Next slide. So we think we can integrate work in Alzheimer's disease and Down syndrome. And what about building a more robust ability to develop, test, and validate effective treatments? The next slide talks a little bit about what is going on here and elsewhere. Next slide, please. Actually, let's go to the next slide to see if it's there. What I was hoping to say is a couple more points, but I can certainly make those. Let's go to these are the folks that really support us, and we thank them very much for all their support. These are the folks in my lab that have done so much excellent work: Qingbiao Wu, Steve Wagner, April Weissmiller, Wei Zhu, Artie Ruparelli, and Matt Pern, prominent among them. Next slide, if there's another one. Maybe not one. So let's go back to the concept now. So now I have very willing cohort of individuals. How do I go forward? Well, the first thing to say is that we focus on APP. And there's several ways of focusing on APP. We could attempt to reduce gene expression for APP. And the thought there is to use these things called ASOs, or antisense oligonucleotides, delivered to the brain of Alzheimer's patients to reduce gene expression. And it sounds a little wild and crazy, but the truth is that it's now a technology being used to improve, reduce the disability, improve the lives of people with Lou Gehrig disease and very soon with Huntington disease. So yes, it's relatively aggressive therapy, but we think it needs to be tested, and we're now working with a company called ISIS to build these antisense oligonucleotides and to try them in the mouse model. A second approach would be to dissolve these plaques. And there's an Elan trial going on right now at UCSD and elsewhere to see if we can reduce plaque buildup. A third approach is to modify the processing of APP to reduce that Abeta peptide to very small sizes, to get rid of those C99 stubs, and to more efficiently generally process APP. And that's a project that is ongoing under an NIH Blueprint grant with Steve Wagner as PI. These are called gamma secretase modulators. In essence, these small molecules make that last enzyme in the pathway work more effectively. And we're very encouraged by what we see there. And then, of course, there's the possibility of using a vaccine. And there we work with AC Immune, again in our mouse model, work that we hope will be published very soon, showing that if we begin that vaccine treatment early, we can actually enhance behavior and prevent degeneration of these cholinergic neurons in the mouse model. So we're encouraged by all those directions for work and by the progress. If we can just go back maybe 3 slides now. And one more. And one more. So let me just then summarize by saying there are fundamental links between Alzheimer's disease and Down syndrome. One more forward. Go back to the one that was just on there would be great. Yeah, good. Let's just stay right there. We think there are fundamental links between really the disease emergence, the mechanisms in Alzheimer's disease and Down syndrome as looked at in humans. We are defining much more clearly now than ever before how it is that those mechanisms work in Down syndrome and in Alzheimer's disease. we think it's now possible to really ask the Down syndrome population to be involved in ways that help us understand the disease, diagnosing it early, and we think there are practical, rational ways in which we can develop and test and validate effective treatments in this population. We see this population, if you will, as pioneers in Alzheimer's disease research, causation and treatment, and this piece of the biology of the domain of Alzheimer's disease as being one that's extremely encouraging and potentially very, very fruitful for all of us. And look forward to more developments in that domain, and thank the Cure Alzheimer's Fund for the support they've provided. We appreciate it very much, and we think, and we're sure it's making a difference in the lives of people with Down syndrome, and also in the lives of people with Alzheimer's disease. So thanks, David. I'm happy to take questions.
And I've got about 1 million of them. So stand by, audience. We'll be talking for the next 12 hours. Now that was fascinating. Thank you for walking us through your research in so much depth, and also so carefully and in a way that that we non-scientists can understand. I really appreciate that, and I know the audience does. So here's some things that occurred to me, and also to our audience, because we have some questions coming up on Facebook. The first question is, you mentioned specifically that there is I think you called it a gene that is an APP gene that some or most or perhaps close to all of the Down syndrome people with Down syndrome have, and the question is would it be that same gene, that same mutation rather, that people without Down syndrome have that also causes Alzheimer's? Or are we talking about a different set of mutations in that population?
All right, good question. So let's break it down in a little detail. So the gene in Down syndrome is normal. It's just that there's one extra copy. So there's no mutation in it. It's a perfectly normal APP gene, the one that you find in you and me and everybody else who's listening. That gene is just fine. It's not mutated. There are families in which a completely normal copy of that gene is present in the absence of Down syndrome. So it's the case in those families that an extra copy of APP is sufficient to cause Alzheimer's disease pathology and dementia. A much larger group of patients have mutations either in APP or in gamma secretase, this last cleaving step. And then there are people with, and certainly the largest group of people with Alzheimer's disease don't have mutations or extra copies of either one of those genes. And presumably what we're dealing in that case are other genes that impact in some way or other the processing of APP, its expression, or the handling of the peptide that's produced. So there are a variety of flavors, if you will, to Alzheimer's disease that all deserve careful attention. But the guess is that there are underlying mechanisms that are very similar, if not identical.
Now, you also mentioned that the population of people with Down syndrome and the families involved and the caregivers involved are obviously highly motivated to participate in research, and they've been very eager and cooperative, I think is close to how you phrase it. One of my questions there is for people who maybe are involved with Down syndrome, have it in their families or caregivers in some sort, but are not involved in research, what steps can they take? Is there a downside to getting involved in research? Is there pain involved? Is there surgery involved? What's the sort of thing on a practical level that a family with Down syndrome how they would get involved in this research?
Yeah, it's a really good question again. So let me just address the idea a little bit. So we would like to see a study done that would allow people with Down syndrome in 30s, 40s, in large numbers, to participate essentially in a natural history study, taking yearly measures of cognitive function and doing non-invasive imaging of their brains together with some blood work. We'd love for those people to allow us to have some spinal fluid, but that would be, that's optional. It's possible to do it. We've had success. Others have had success with that. But in the main, this is a very non-invasive collection of data at intervals to map out, if you will, the natural history of Alzheimer's disease and Down syndrome. Happily, we have a pilot project underway right now with only 12 patients, but we're learning a lot. And the NIA has just announced a larger project that will allow more patients to be enrolled, and I'm sure a number of institutions will apply for that application. We'll certainly be one of them. It's very helpful that we've collected data already, and we kind of know what kinds of things are possible with this population and what kinds of things are not possible. For people who are interested in this, it would be very useful for them to let us know at UCSD that they are interested. And what we could do is to make, if they can tell us where they are, we can try to help find local clinical trials units in academic institutions to enroll them, to engage them, and to make it possible for them to participate in research. There's no ready way to do that right now, but I think we're happy for any volunteers. We have a very active Alzheimer's disease, adult Down syndrome clinic here at UCSD. We'd be delighted to see any and everyone here. We're building our adult Down syndrome group very, really rapidly, so we'd happily see anyone here. But we'd also be very happy to try to find other institutions where they might more readily geographically enroll and be involved in what we think needs to be a very large natural history study that will require large numbers of people.
So there's an option for invasive but also non-invasive research, as you said. And is there an 800 number or something? I know people could get in touch with Cure Alzheimer's Fund, and we could pass on the information. People could obviously find you on the web. Is there some kind of more direct number or address that you'd like to leave?
I think if they could go through your office, it would be great. And then what we can do is, actually what I'll do today is I'll send you a link for our Down Syndrome Center. Sure. But if they could go through CAF, that would be wonderful. I would really appreciate that. I really wish I had that website name right in front of me, but I don't.
Well, it's all right. We're easy to find on Facebook, and we can certainly pass along information. Great. So let's stay on this thread, because I'm trying to think, as I'm trying to put myself in the position of a caregiver, someone in the family of someone with Down syndrome, and let's address directly. I think the sensitivity and one thought might be, well, so you know, those in the Alzheimer's community are kind of grabbing the Down syndrome people as guinea pigs to help the Alzheimer's people. But what is Down syndrome? What does the Down syndrome community get out of that? So perhaps you could address that, and we could talk about it. You know, just kind of face that question head on. There's obviously, we don't want anyone to think that we are taking advantage of them. And as I understand this, the research that you're doing stands to benefit both patients with people with Down syndrome, but also people without Down syndrome that are facing Alzheimer's disease.
Well, that's good, and I'm aware of the sensitivities, and we've encountered that as we built our adult Down syndrome clinic. It's pretty clear to me that the first people to benefit from what we do will be people with Down syndrome. We, I think, have done a good job of stressing the benefits of studying Down syndrome to the rest of the world interested in Alzheimer's disease. And we're happy in a way that we can, that the visibility we have is increased substantially by the fact that there is such a thing as Alzheimer's disease in the general population. But the work we do in the lab and the work we do in our clinic is first and foremost to benefit people with Down syndrome. If you will, to allow the next generation to be able to say, people with Down syndrome used to have to deal with the inevitable onset of Alzheimer's disease, and that's simply not true anymore. Research was done across the world that changed the game for people with Down syndrome. That's the first objective for us. And we're delighted to think that we can help everybody else. And we're delighted to think that these most vulnerable people, by helping themselves, will help others. But I would argue just very clearly that the benefits are first and foremost focused squarely on people with Down syndrome.
And thank you for clarifying that. I think this is my final question. You mentioned several different things that are going on that's kind of the next phase for you, and you listed a lot of research. Some of those people are directly getting research money from Cure Alzheimer's Fund, which is very exciting. Perhaps you could talk about, I'm just going to ask you to kind of double back and talk about what is the most exciting thing going on right now, whether it's funded by Cure Alzheimer's Fund or not. What is the most exciting thing that's going on in this area, and whether it's the most promising, but something that we can get excited. I know that you and a lot of people in this area are just really excited about something that you know, potentially some real breakthroughs maybe in the next couple of years. What could we see if all the ducks line up there in a row and we get lucky and this comes together?
The one most exciting thing, and it's exciting not just in its conception but in the success we've had, is work under Steve Wagner, where we've developed with NIH funding and with a great deal of help from advisors several small molecules that get in the brain and then act in the brain to enhance the processing of APP. And by that I mean these stubs, these C99 molecules that we know are trouble, and the Abeta peptides that we know are trouble, are being reduced to tiny levels, almost to levels that are normal in brain experiments, in animals, in mice, in our mice, the mouse model of Down syndrome. We're so excited about that because here's the excitement. We know it's APP. We know what part of APP it is, and these small molecules target exactly the culprits that we've identified in our studies. So that's very exciting. And the idea that we've gotten this far with molecules that are this potent and this brain available, that are as effective as this in engaging APP processing, is extremely encouraging. And you know, I'm hoping that we're talking about just a few years and being in phase 1 trials. Of course, one can't promise that. All kinds of things there are all kinds of bumps along the road. By the way, I was never so aware of that as before I came involved in this project. I mean, literally anything that can go wrong may go wrong with any compound. But so far so good, and we're going to persevere because we think this approach is exactly the right one, and we're very grateful for the chance to work in this area, and we're encouraged.
Well, I just can't thank you enough for your personal commitment to this line of research. I can say on behalf of the Alzheimer's community, and I don't have a personal connection to the Down syndrome community, but I will say on behalf of them too, it's obvious how much intelligence and energy is going into this, not just on your behalf, but on behalf of all the researchers that you listed. Thank you so much for your now lifelong commitment to this cause, and for taking the time today to explain it to us. I could go on and on with many questions, but we're out of time. So let me just say a few words to end the program. That's all the time, unfortunately, we have for our program today. Thank you, audience, for watching. If you have any further questions about the science in today's show, please do post them on our Facebook page at facebook.com/curealzheimers. Inside the Alzheimer's Lab is a production of Cure Alzheimer's Fund, a not-for-profit dedicated to supporting the research that will stop this terrible disease. Every dollar you donate to Cure Alzheimer's Fund goes directly to scientific research. Its founders cover all administrative costs. That's an amazing thing. And just to repeat that, you give $1 to Cure Alzheimer's Fund, that is 100 cents that will go to an Alzheimer's lab like Dr. Mobley's for research. The founders of Cure Alzheimer's Fund are Jeff Morby, Jackie Morby, Henry McCants, and Phyllis Rappaport. Thanks again to our guest, Dr. William Mobley. I'm David Shank. Thank you so much for joining us.